will be Dr. Dalin. Okay? He's already connected? Excellent, excellent. He's professor of the marine biochemistry at the State Key Laboratory of Marine Environmental Science in Xiamen University, China. His research interest is integrate lab experiment with field studies in the natural communities. For example, the interaction between phytoplankton ecophysiology and nutrients biochemistry, the role of the trace metal in marine biochemistry cycles of carbon and nitrogen, the response of phytoplankton to the global change, nitrogen fixation, and oligotrophic marine ecosystem. We welcome to Dr. Dalin.
Hello, everyone. First of all, my apologies for not being able to attend the symposium and give this presentation in person due to COVID-19. Truly sorry. But I would like to take this opportunity to thank the symposium's organizers for inviting me to speak here. It's truly a privilege. So the title of my talk today is The Complex Effects of Ocean Acidification on Marine Phytoplankton in a Multi-Stressor Environment. We know biological impacts of OA have been a focus of oceanography and marine biogeochemistry studies for more than 2 decades. However, after so many years, there is still a lot that remains to be fully understood on this topic. The more we study it, the more we find out how complex it is. So, understanding the complex effects of OA on marine phytoplankton in a multi-stressor environment is really challenging. So it's giving a talk on this topic, I would say. So let's get started. We are all familiar with ocean acidification, and we all know that oftentimes OA refers to a decrease in seawater pH as the ocean takes up anthropogenic carbon. But personally, I would prefer what Scardoni defines in this review article, that OA means the addition of carbon dioxide in seawater that causes a reduction in ocean pH and shifts in carbonate speciation. So what I would like to emphasize here is that when studying biological impacts of OA, all these 3 parameters should be taken into account. Over the past years, there are numerous studies examining biological effects of OA on a variety of marine organisms, and my talk today focuses on marine phytoplankton. As primary producers in the ocean, they fix CO2 into organic matter, contribute about half of global primary production, and more importantly, drive the marine biological pump. Also, they serve as the base of marine food webs. Therefore, it is very important to understand and predict the effects of OA on marine phytoplankton. Specifically, for today's talk, I will introduce studies mainly on diatoms, picocyanol, including Procococus and Synechococcus, and nitrogen fixers. They all play an important role in ocean ecosystems. 2 key questions we focus on in this talk are, first, by what mechanisms will OA, including an increase in CO2 and a decrease in pH and carbonate, affect phytoplankton's physiology? And the second question is, how other concurrent and complex stressors in association with global change will modulate OA effects on phytoplankton. These complex stressors, including like solar radiation, nutrient condition, temperature, and so on. This is the outline of my talk. First, I will focus on effects of OA on marine phytoplankton, which includes the individual and combined effects of CO2, pH, and carbonate. Actually, this is already very complex, but only a mechanistic understanding of these effects can help us better understand the interactive effects of OA and other multi-stressors. And at the end, I will provide some perspectives in terms of challenges and strategies for multi-stressor studies. So first, effects of OA. As I just mentioned, OA effects on marine phytoplankton are complex because increasing CO2, decreasing carbon and pH individually can affect phytoplankton by different mechanisms. First, let's take a look at increasing CO2. We know in photosynthesis, the light reaction provides energy and electrons for the dark reaction, while CO2 is fixed into organic matter by the enzyme rubisco. Rubisco is a very ancient enzyme, and according to some people, it is the most abundant protein in the world. Why it is so abundant? Because it is a very lousy enzyme with a very poor affinity for its substrate, CO2. As you can see from this graph here, that the half saturation constants for CO2 fixation of rubisco in all these phytoplankton groups are higher than the typical CO2 concentration in surface seawater. In other words, the enzyme is undersaturated with the current level of carbon dioxide in seawater. So marine phytoplankton are potentially carbon limited. To get around this, most phytoplankton operate the so-called carbon concentrating mechanisms, CCMs, which elevates CO2 concentration intracellularly at the site of carbon fixation by rubisco. For example, outside 10 micromolar, inside here over 100 micromolar. And this is not trivial, because in fact, the membrane is very permeable to CO2. So you pump it in, but it keeps leaking out, just like a leaky bucket pouring water into it. It's a lot of work to do that. Therefore, it is expected fully that if you increase CO2, less energy will be spent in pumping carbon, and we will see an increase in growth. But this is what we find. Overall, we don't see a systematic and significant increase in photosynthesis under high CO2, very noisy data, and not very convincing OA effects. Why? Well, one of the main reasons is that the energy for concentrating carbons is far less than the energy for photosynthesis, and this has been shown in the diatom Phytoplankton trichonewton and Protococcus. Carbonate. We all know that OA will lead to a decrease in calcium carbonate saturation state in seawater, thereby affecting calcification. However, we found that there is not a very systematic set of data on the OA effects on calcification in coccolithophore. This could be due to different experimental conditions among studies or due to species-specific responses. And we should keep in mind that calcium carbonate is formed intracellularly, and different calcifiers may have different capacities for regulating internal environments in response to OA. Besides affecting calcification, a recent study shows that decreasing CO2 can affect ion uptake in diatoms. A protein in diatoms called ISIP2A, which is a ferriion concentrating protein, is actually a phytotransferrin. It can use carbonate to bind ion and then transfer it into diatom cells. So this is an ion uptake system that is sensitive to changes in carbonate concentration. As you can see from the figure here, a decrease in pH causes changes in both carbonate and ion. Consequently, ion uptake rate decreases, and part of this is attributed to the influence of decreasing carbonate at low pH. How about pH? pH can have various effects on marine phytoplankton. For example, it can affect functions of key enzymes. This slide here shows the pH effect on the activity of alkaline phosphatase, which is an enzyme that allows phytoplankton to use organic phosphate when they are in short supply of inorganic phosphorus. Another example for pH effects on enzyme function is nitrogenase in the globally important antofixing cyanobacteria trichodesmin. We can see here, low pH lead to a decrease in nitrogen fixation rate, but strikingly, the abundance of the enzyme nitrogenase increase at low pH. So more enzyme, but with slower nitrogen fixation rate, which means decreased nitrogenase efficiency. Why? This is because nitrogenase is also a hydrogenase. It fixes nitrogen into ammonia and meanwhile produces hydrogen. In acidification, it has been shown that the pH can affect the distribution of electrons between nitrogen fixation and H2 production. In this case here, as pH decreases, more electrons are used to form hydrogen production. And indeed, we see in trichodesmine an increase in H2 production at low pH regardless of CO2 concentration. Low pH can also affect intracellular pH homeostasis in phytoplankton. And this is what we found again in trichodesmine, that acidification resulted in low cytosolic pH. In an attempt to maintain pH homeostasis, trichodesmine upregulated several proteins involved in proton pumping and energy production for maintaining pH homeostasis. In Dalton's, low pH resulted in significantly decreased silica production, and this is likely due to enhanced silica efflux from the Dalton cell at low pH, but not caused by altered growth or cell size. In addition to physiological effects, pH can also affect phytoplankton chemically by changing chemical speciation of key nutrients. We know solubility of particulate iron should increase at low pH, but for dissolved iron, its seawater chemistry is very complex and also very sensitive to pH change. For example, low pH was found to increase concentration and half-life of iron 2, which should increase iron availability. But on the other hand, low pH can lead to increased complexation by organic ligands, thereby decreasing iron availability. So, very complex. Here is another example for low pH effects on chemical speciation of key nutrients. pH can change the chemical equilibrium between ammonia and ammonium. As a result, ammonia oxidation rate would decline at low pH, as shown in this study. So far we have seen that OA effects on marine phytoplankton are really complex, because each one of these three parameters can have different separate effects. But what makes it even more complex is the interactive effects of these three parameters, which could be additive, synergistic, or antagonistic. This is a recent study on the Dalton turtle series. We investigated how the Dalton was affected by CO2 and pH, both individually and jointly. We found that CCM downregulation was attributed to carbonation, and photosynthetic carbon fixation, as well as CO2 contribution to inorganic carbon uptake, increased, mainly due to acidification. So acidification can play a vital role. Together, CO2 and pH can lead to an additive positive effects on this Dalton. Another example for the interplay between CO2 and pH that I would like to show is on trichodesmine. Here we see OA lead to a decrease in both growth and nitrogen fixation rates. When we separate CO2 and pH effects, we can see, in fact, high CO2 had positive effects, whereas low pH had negative effects. The positive effects of high CO2 was due to CCM downregulation, as we can see here, the increased CO2 affinity at high CO2. And the negative effects of low pH include reduced nitrogenase efficiency, which we mentioned a while ago, and increased cost for maintaining cellular pH homeostasis. Because the positive CO2 effects were overwhelmed by the negative pH effects. As a result, the net effect of OA is decrease in growth and nitrogen fixation rates. A quick summary. So far, we have seen that OA effects on marine phytoplankton are really complex. This is because CO2, pH, and carbonate can have separate effects by various chemical or biological mechanisms. And on top of that, the interactions between them can be additive, synergistic, or antagonistic. Now, let's move forward to the interactive effects of OA and other multi-stressors. And this is even more complex because different marine ecosystems have different chemical and physical dynamics in terms of OA and other stressors. First, let's look at the carbonate chemistry. We can see here that short-term pH dynamics differs in different marine ecosystems. It is generally stable in tropical ocean and varied very little at high latitude, but in coastal and estuary environments, it's much more dynamic. In decadal and longer time scale, in high latitude oceans, acidification shows large and rapid rates. In tropical and subtropical oceans, like beds and huts, we see a stable pH decline, generally reflecting the increase in atmospheric CO2 due to the anthropogenic activities. But in coastal waters, in some cases, there appear to be pH decline, but overall, it's much more dynamic with complicated controls by ocean physics, chemistry, and biology. The complexity with studying OA effects is that it's not happening all by itself. Other environmental factors are also changing in the ocean. For example, warming and mixing, and as a result of warming, water column stratification will intensify, which will reduce nutrients input from the bottom and increase the average light intensity for phytoplankton. So, within the context of global change, in diverse marine environments, OA dynamics can be different. And on top of that, there are other stressors with regional and local characteristics. In high latitude, rapid OA, intensive warming, increased irradiance, and ion limitation may intensify or lessen. In tropical and subtropical ocean, it's likely nitrogen will become more limiting for phytoplankton, and for diatotrophs, the limiting nutrient will be iron and or phosphorus. The coastal ocean is a much more complex system. The environmental conditions here are very dynamic, and there are many stressors. So for today's talk, I will mainly focus on these two scenarios, the high latitude and tropical subtropical oceans. For each of these two scenarios, I will give a few examples to showcase how other stressors may modify OA effects on phytoplankton. For studies on the coastal ocean scenario, I will only introduce it briefly, but they are important as well. So first, high latitude ocean. Here shows two studies conducted in the HNLC regions. One is in the Bering Sea, and the other is in the Weddell Sea. First, both studies indicate that OA will lead to an intensified ion limitation. Second, they both show that when iron was limited, there was not much effect of OA on growth and phytoplankton community composition. Only when iron was added, we can see an increase in primary production and a shift in community structure. So these studies indicate that iron is primary control, and iron availability can modulate OA effects. In high latitude HNLC regions, phytoplankton is often limited not only by iron, but also by light. In this study with the model delta PT, we found that OA lead to increased growth rate and carbon fixation rate, especially under iron and light co-limitation. This is because under iron and light co-limited conditions, energy production is severely impaired, So in this case, the energetic savings of CCM downregulation by high CO2 is more beneficial for the cell. And high CO2 also provides a large electron sink to facilitate electron transport, thereby elevating oxidative stress. So this study showing here also indicate that OA effects can be modulated by iron and light levels. But different from what we observed with PT, which is a temperate species, there is a lack of high CO2 effects on growth of thousand ocean diatoms. For example, in these 2,000 ocean diatoms, although CCM were indeed downregulated at high CO2, growth rates were generally unaffected. This is likely due to the relative lower energy requirement of the CCM at lower temperature, because we know at low temperature, the CO2 solubility in seawater is higher, and the half saturation constant for CO2 fixation of Rubisco is lower. Tropical and subtropical oceans. This is Kunshan Gao's work. They found that OA lead to decreased instead of increased primary production in diatom-dominated natural assemblage at several stations in the South China Sea. Further lab experiments with the model species revealed that OA effects on diatoms can be modulated by light intensity. At low light, high CO2 cells had a higher light use efficiency. Therefore, OA lead to increased growth rate, whereas at high light, high CO2 decrease quantum yield and increase the light stress, so OA lead to decreased growth rate. Synechococcus and Prochlorococcus are the most abundant phytoplankton in the tropical and subtropical ocean. These early studies by Fu et al. Found that in Synechococcus, OA lead to slightly but not significantly increased growth rate, and the growth rate increased the most under high temperature and OA conditions, the so-called greenhouse scenario. But in Prochlorococcus, growth rate remained unaffected by increasing either temperature or CO2. So far, most field studies on OA effects on nature phytoplankton communities are conducted at high latitude and coastal regions. So over the past few years, we investigated the response of phytoplankton-dominated community to OA in the tropical and subtropical western North Pacific. We found that although cyanobacteria accounted for more than 90% of the community according to pigment analysis, it was eukaryotics that dominated the response of primary production to OA. And there was a positive correlation between OA-induced changes in primary production and in situ chlorophyll A level. We hypothesized that this switch from stronger negative OA effects at low chlorophyll to less negative effects at relatively higher chlorophyll levels was likely related to changes in nutrient availability across this study area. And this was supported by nutrient amendment experiments, which show that nitrogen and phosphorus addition offset the negative effects of OA on primary production. So this study here indicates that OA effects can be modulated by nitrogen and phosphorus levels. Besides nitrogen and phosphorus, growth of cyanobacteria can also be limited by iron. In this lab study, we found that OA stimulated the growth of iron-limited Synechococcus, whereas it had no effect on growth under iron-replete conditions. This is because cyanobacteria, as we know, its CCM is very efficient. So under iron-replete conditions, although the CCM was indeed downregulated at high CO2, there was not much beneficial effects. By contrast, in iron-limited cells, iron-containing photosystems were downregulated and ROS levels were high. So in this case, high CO2 elevated oxidative stress, which could save iron and use it for photosynthesis. Therefore, OA lead to increased growth rate in iron-limited cells. For Proctococcus, we studied the interactive effects of iron, light, and OA, and we find that increased light intensity enhanced OA effects under iron-limited conditions, and again, no effect was observed under iron-replete conditions in terms of high CO2. The main mechanisms underlying this observed effect is related to anti-oxidative stress. We found that in these low-light adapted strains of Proctococcus, oxidative stress increased with increasing light intensity, and iron limitation further increased the level of oxidative stress. Therefore, under such conditions, OA greatly elevated oxidative stress caused by high light and low iron, and as a result, the growth rate increased. For tropical and subtropical ocean, a particular example I would like to show is on the N2-fixing cyanobacteria, trichodesmin. They are an important source of new nitrogen for the oligotrophic marine ecosystem, where they are often limited by iron and or phosphorus. We found that OA has negative effects on both the growth and N2 fixation rates of trichodesmin, and iron limitation can amplify the negative effects of OA. This is because acidification leads to low cytosolic pH and reduced nitrogen fixation rate. So to cope with this, on one hand, trichodesmin synthesizes more nitrogenase to compensate for the reduced nitrogen fixation rate. On the other hand, they upregulated several proteins involved in proton pumping and energy production in order to maintain pH homeostasis, and both of these require iron. So, under iron-limited conditions, trichodesmin reallocated iron from these iron-containing proteins to nitrogenase. As a result, both proton pumping and energy productions were affected. Therefore, growth and N2 fixation further decreased. In the oligotrophic ocean, trichodesmin can also be limited by phosphorus. So to mimic P limitation, we use chemostat to investigate OA effects on phosphate-limited trichodesmin. We can see here that after about 10 days, the culture reached a steady-state P limitation. pH levels were well maintained at about 8.1 and 7.8, and phosphate concentration was kept at low, about 15 nanomolar, which is close to typical DIP concentration in open ocean surface waters. We found that carbon and nitrogen fixation rates both decreased under OA. Interestingly, we also found that P requirements increased under OA. We are looking into this and think it may have to do with accumulation of poly-P, likely for maintaining cytosolic pH. Besides laboratory mechanistic studies, we also went to the field to examine how nature populations of trichodesmin will respond to OA and whether iron and phosphorus can modulate the OA effect. Here shows the result of 10 incubation experiments we did with trichodesmin-dominated diatrope community in the South China Sea. In 9 out of 10 experiments, nitrogen fixation rate decreased by 5 to 73% under acidified conditions. At 2 stations, we also did nutrient addition experiments, which show that trichodesmin populations at these 2 stations were limited by iron and P respectively, as indicated by the expression of iron and phosphorus limitation marker genes. And with iron and phosphorus additions, the negative effects of OA diminished. So this field data also suggests that iron and phosphorus limitation can amplify negative effects of OA. We did similar work with diatrial communities in the North Pacific subtropical gyre. In this region, UCYMB was the dominant N2 fixer. So we also find the on average nitrogen fixation rates decreased by 36%. And nutrient addition experiments also show that iron limitation amplified negative effects of OA. Last but not the least, let's see a couple of examples for coastal oceans. We know that carbonate chemistry is very dynamic in coastal waters, but so far there are not many studies that apply a fracturating carbonate chemistry regime to study OA effects on coastal phytoplankton. In this study shown here, the authors used both fracturating and steady regimes of carbonate chemistry to compare OA effects on coastal and oceanic diatoms. They found that the coastal and oceanic species differed in the ability to cope with dynamic carbonate system. The oceanic species was negatively affected by the fracturating regime, whereas the coastal species showed no difference in their response to the two carbonate chemistry regimes. In coastal oceans, Wei Junkai's study has shown that acidification of subsurface waters can be enhanced by eutrophication, and this has also been observed in coastal waters of China, for example, the Bohai Sea in the summer. Although shellfish are not the focus of this talk, such eutrophication-enhanced coastal acidification will present a big challenge for shellfish aquaculture business. Take China, for example. It's the world's #1 country with 84% of global shellfish production. However, at the moment, there is still a lack of monitoring and lack of awareness of OA in China's coastal shellfish farms. Besides stressors like warming, changes in light intensity, and eutrophication that we just talked about, there are other stressors in coastal regions, for example, pollution, hypoxia, and so on. So coastal ocean is truly a very, very complex system and definitely warrants more studies because the coastal ocean is so important that it provides many ecological and economic services. So the last part, let's look at the challenges and strategies for multi-stressor studies. So, challenges for studying OA effects in a multi-stressor environment. First, there is a wide range of stressors that either individually or interactively can modify both the effects and severity of OA through a variety of pathways. Second, the co-occurrence of stressors, as well as the magnitude and direction of their combined effects, vary across space, time, and organization levels. Third, effects of multi-stressors are often non-additive, so the design and interpretation of multi-stressor research is really complex. Last, all research approaches, including like manipulation, incubation experiments, nature gradient experiments, and so on, they all have benefits and limitations, so there is no single ideal method. I should also mention that my talk today focuses on multi-stressors, so I didn't include adaptation, acclimation, and evolution aspect of OA study, but I should say they are also very important. Boy et al. Developed a strategy that used a tractable experimental design for multi-stressor research. First, they identify and quantify all of the key stressors relevant to the research questions. And then an idealized full factory design defining all of the stressors and a range of interest for each one is identified. Last, they identify the most relevant subset and levels of stressors to create a collapsed design that best addresses the questions of interest. For example, in this multi-stressor study on the subantarctic diatom Pseudonychia, temperature was identified as the dominant stressor, and OA, nutrients, iron, and light together was a collapsed non-dominant stressor. These two stressors, along with their interplay, can explain more than 90% of the observed physiological changes. In particular, they found enhanced growth was mainly owing to warming and ion enrichment. A holistic approach based on examining limiting resource can also be useful to multi-stressor studies. This approach takes a holistic view of metabolism by examining the way in which environmental changes alters the use of limiting energy or materials. It offers a common framework for handling the diverse traits, life history characteristics, and metabolic capacity of different organisms. Here shows an example for a diatom cellular energy budget. So the budget is constructed based on mechanistic and quantitative understanding of key cellular processes involved in energy generation, like photosynthesis, energy expenditures, like carbon fixation, CCM, nitrogen assimilation, and so on, and energy storage, for example, like carbohydrate, lipid, and proteins. So, by quantifying and incorporating energy changes associated with changes of these key processes in response to multi-stressors, in this case, light, nitrogen source, and OA, the energy budget did a reasonable job predicting the effect of multi-stressors on photosynthesis. This is another example for the holistic approach based on examining limiting resource. It is an optimality-based trichodesmine cellular model. So the model is constructed based on mechanistic understanding of cellular physiology, and it has a solution for optimal allocation of intracellular energy and ion for maximal growth at a given condition of OA and ion, and we can see the model well reproduces the measured growth rate. So, we know that there is no single ideal approach to study multi-stressor effects. Therefore, integrating laboratory, field, and modeling approach is definitely a strategy, but it is also a challenge at the same time. In models like the Earth system model, the ecological imprint of climate change is a slow chain overlaying internal variability on time scales from episodic events to seasonal cycle and interannual climate mode. But lab and field perturbation experiments use batch culture approaches that could only mimic an episodic event such as atmospheric deposition. So how applicable are the transient outcomes obtained by lab and field mesocosm experiments to seasonal cycles? It is really a challenge. So improving the links between model and experimental designs is needed for the future multi-stressor studies. Some take-home messages. Understanding OA effects on marine phytoplankton in a multi-stressor environment requires observation and experiments with local and regional characteristics of ocean physical and chemical dynamics, but within global context. Observation on spatial and temporal patterns of OA and other stressors, as well as their interactions, are essential for the design and finding interpretation of multi-stressor studies. Identifying mechanisms by which multi-stressors affect marine phytoplankton is critical for understanding and predicting OA effects with the real-world complexity. Based on observation and mechanistic understanding, continued development of experimental design and approaches is highly encouraged. Last, multi-stressor effects on phytoplankton can only be understood by an integration of observation, mechanistic and scenario-oriented study and modeling, which itself presents challenges. Taking this opportunity, I would like to thank all my group members and my collaborators around the world. And thank you very much for listening. Again, my apologies for not being able to present in person. As we have seen from today's talk, OA effects on marine phytoplankton in a multi-stressor environment is truly complex, and studying it is a real challenge. So I would be very happy to hear any questions, comments, or suggestions from you all. Thank you.
Okay, well, we thanks Dr. Dalin. He just was connected. We really appreciate his presentation. Nice presentation. Well, in this way, we finish with the last invited, the last speaker. And it's amazing because I just feel that we just started. We have a very intense days. We started in weekend, some of the people that are here in the room, with several sites events. There are one more today after this ceremony. But several conclusions we can get from this experience, the after and before the COVID. And one of the analyses I was thinking was that you must know that this is the first symposium hybrid, and this is a challenge. Probably more expensive, but probably very convenient. We will see. We need to do the analysis, right? The number that I have in person was 185 people was here, and virtual was 90. But I don't know the numbers that the people that was connected. Probably we will know it by YouTube, maybe, with the records on that. And I'm talking about that because maybe we can further in this kind of experience. The other thing is, in Mexico, we have the experience to be working with the symposiums like this one, but just virtual. The talk was recorded, and we can store it as a library for the future, and which is kind of convenient, right? Then the people, the local, the international members need to be together to rest first, of course, and then to get the conclusions. And in these ways, I was just to say thank you to everybody because these kind of symposiums are alive because we are here. And we need to be very pleased that we are presential and to see the faces to everybody, to say hello, to give a hopes. And then so pleased that we see you, several people I just met virtually, and here I was looking that was taller than me. Many surprised many people, right? It was really nice. Okay, and I'm going to share the land to Michelle and Jorge Chaname to follow with the program just for the end.
Buenas tardes. Llegamos al final. Realmente muy contentos de haber podido lograr esta tarea, esta jornada mundial con todos ustedes acá en el Perú, en Lima, Perú. Cuánto hubiéramos querido que hubieran estado en Lambayeque, pero la logística tal vez no hubiera podido ayudar todas las comodidades que han tenido acá en Lima. Pero creo que será un gran recuerdo de este quinto simposio acá en Perú. Y gracias por la confianza depositada en nosotros en una universidad nacional del norte del país, gracias a la gestión que realizaron el doctor Wilmer Carvajal en su momento, y como les decía, mis colegas sobrevivientes de la pandemia, teníamos que hacer algo y teníamos que seguir para adelante y honrar nuestros compromisos adquiridos antes de la pandemia. Creo que el evento de esta manera híbrida, y creo que la presencia, el calor humano de estar juntos después de cuánto tiempo, de haber realizado reuniones en el MIT, como lo decía el colega Martín, ¿no? El encontrarse, qué alegría, ¿no? De verse después de cuánto tiempo, colegas de todo el mundo que han estado conectados, pero no había esa presencia que es la condición humana de el abrazo, del
closeness. I think beyond all the academic part, the scientific part, the human warmth, the being able to gather and share your breakthroughs and accomplishments, it gives us much pleasure after so much time. And I would like to thank, honestly, all the institutions who supported us this week so that this symposium would take place in the best conditions possible. Beyond some small details that might have caused some discomfort, I think, in general, we were all very happy with this exchange of ideas and proposals, where once again we see that there's still a lot of work to be done to incorporate the main actors, which are the fishing communities that dedicate their lives to the sea, and our heads of state, our politicians, the decision makers, who sometimes don't help us that much with the protection of the ocean. And my thanks to all the institutions who made this possible, and especially the Principality of Monaco, the Foundation, the institutions. I can't memorize all of them, unfortunately. There are so many here. The institutions that bet on us to be able to perform this event. Honestly, I'm so happy. My bosses, the dean and vice dean of research, and this makes me happy. Also, that the work and the backup of the university to make this event possible was worth it. So I would like to thank, honestly, all the people who came here all week. Maybe there were some inconveniences, but to get to know Dr. Gattuso, Dr. True, I mean the last stage, and all the weeks we were there, moving forward even with the language limitations, working to be able to achieve this objective. And I can't thank Michel enough, because he was there from the beginning and was trying to assume this great responsibility. Let's say the representatives, they were not the direct